Okay, let's talk about a huge problem in cancer research, finding good biomarkers for CDK4/6 inhibitors.
These drugs are a game changer for breast cancer, but a major puzzle is holding back more progress.
So why can't we find reliable markers for them? Our big screens just keep giving us conflicting results.
This has been a huge roadblock. Data from major screens like GDSC2 and PRISM is just all over the place.
Well, it turns out the answer might be in a really basic flaw: how we're measuring things.
It all boils down to how we measure cell growth. Are we using metabolic assays or DNA-based ones?
See, metabolic assays don't actually count cells. They just measure things like ATP as a stand-in for growth.
And this gets to the heart of the problem. What happens when cells stop dividing but keep on growing?
CDK4/6 inhibitors slam the brakes on the cell cycle. Basically, they tell cancer cells stop dividing.
But here's the twist. The cells stop dividing, but they don't stop growing. They just get bigger.
And as they get bigger, they need more power, so they start churning out more mitochondria.
More mitochondria means more ATP, and that creates a totally false signal of high metabolic activity.
And here's the proof. DNA assays correctly show the arrest, but the ATP assays, they completely miss it.
So is this real drug resistance, or is it just a massive measurement error?
So what happens if we only look at the data from the reliable DNA-based assays? When you do that, clear genetic dependencies pop right out from the DepMap data. It all starts to make sense. Suddenly, you see it. Sensitive cells need the drug's targets. Resistant cells, they've found a workaround.
And this points to real biomarkers. High RB1 might mean sensitivity, while high cyclin A1 could mean resistance.
But get this, none of these clear signals show up in that messy metabolic assay data. They're totally hidden.
And this isn't just about CDK4/6 inhibitors. The implications here are huge for a lot of drugs.
Think about it. Any anti-cancer drug that arrests the cell cycle could be giving us these same bad readings.
So the authors make a powerful point. We need to rescreen these drugs using the right kind of assay.